The overall goal of this procedure is to perform targeted cell ablation of kidney cells in the zebrafish embryonic kidney, the pronephros, for the purpose of studying renal epithelial regeneration following acute kidney injury. This is accomplished by first microinjecting zebrafish embryos with a dextran fluorescent conjugate. During an overnight incubation, the conjugates are selectively absorbed by nephron proximal tubule cells. The next day, the cells are examined under epifluorescence and selected cells are ablated using a pulsed laser system. Later, morphological and molecular changes are assessed using histology, whole mount in situ hybridization, and immunohistochemistry.
The main advantage of this technique over existing methods, like acute kidney injury using the nephrotoxic antibiotic gentamicin, is that such treatments trigger catastrophic damage and are lethal, precluding the analysis of renal regeneration events over time.
This method can help answer key questions in the kidney field, such as the identification of molecular signaling events that occur during epithelial regeneration. We first had the idea for this technique when we wanted to develop a detailed analysis of renal epithelial regeneration.
In petri dishes, collect fertilized zebrafish embryos in modified E3 solution without methylene blue. Raise the fertilized embryos at 28.5 degrees Celsius until they are between 48 and 55 hours post-fertilization, when the embryo's renal system has begun to function.